This is a production of Cornell University.
And so, yeah, it is a real pleasure to introduce Travis Wrightsman for his defense. He was born and raised in LA, and so he and my wife have always connected over being Southern Californians and things like that. And so, but he did his bachelor's at UC Riverside and in biochemistry. Got started in understanding in his world of understanding bioinformatics and genomics, working with Sue Wessler, who was a incredibly important figure in everybody in May's genetics today. But he worked with her to start really annotating transposable elements in the citrus genome. And I think really started forming his ideas on how bioinformatics should be done, how it should be shared with the community and everything. He then applied to Cornell, got in here. We were excited to have him, but he also got a Fulbright all at the same time. And so he said, Ed, I don't want to start here. I'm going to go to Germany. So he went to Germany for a year and worked with Detlef Weigel's group. And there he helped develop a pipeline of automated assembly and annotation of Arabidopsis genomes. That was really important for understanding the strengths and weaknesses of graphs at the time. He then joined us in 2018, and he's going to be telling you about his research there. But as part of that, he also took some time off to work with Google X, and he's wearing his Google X T-shirt today. But I think you'll see that there's a very strong streak in Travis of both public sector commitment, but also wanting to understand how industry has worked in putting those two together. He's going to go back to Germany to do a postdoc in bioinformatics, and yeah, I'm sure he'll do great things. But every graduate student brings their strengths and weaknesses to graduate school. But what I really want to highlight about Travis is this perspective he has on science. And he was one of the graduate students, I would argue, that has thought more deeply about how science and technology should be developed. And he really was asking fundamental questions about what does it mean to conduct an experiment? How should experiments be repeated? How does one share and repeat these experimental findings? And then, what are really the strengths and weaknesses of public sector versus private sector science? And I was Travis, I was thinking about it last night. You're kind of like a science philosopher, okay? Who thinks about some of these things about how do we do science and make it repeatable. But he's also has that blue collar perspective of getting things done, like plant breeders need to get things done. And it's that combination of things that's been really wonderful. And so he's really been a leader, not only in our lab, but across the community in establishing rigorous methods for designing and setting up scientific computational pipelines so that our science can be repeatable. And he's combined that with knowledge of really the front of machine learning, whether here or working with Google X. And although we tease Travis about his smartphone, which he has an American-made open source smartphone, which probably took him about 6 months to make it so he could take a picture or something like that. I mean, longer. Okay. I mean, it took a year before he could get the thing to take a picture. Okay. But it really emphasizes the way he tackles problems. He wants to understand how things work and work on systems that other people can participate in. And I think it's as machine learning and AI become really even more distant and unrelatable to people, I think Travis is really there trying to make sure that we're doing science and apply these incredible, powerful tools in a way that's democratizing and really available to us. So, Travis, take it away and tell us all about how genes work and what we can do with that knowledge.
All right. Well, thank you very much, Ed. And for the record, if I allowed my phone to install Zoom, I would definitely be presenting from it today. But regardless, that's not what you're here to listen to me talk about, but rather the culmination of my PhD work that I've titled From Arabidopsis to Zea, learning conserved cis mechanisms of gene regulation. Classic. So my PhD has been broadly focused on these two research questions. The first is, where are the causal loci in cis regulatory regions? And second, how do we transfer knowledge across species? And I'm talking to a breeding audience. You might be wondering why I'm mentioning causal loci. And the reality is that if you want to do efficient genome editing, you need to know where the causal loci are to edit them. And genome editing is just the latest methodology in the massive toolbox that plant breeders have to make genetic gain. So to borrow Ed's software analogy, we can see that breeding methodologies have gone through sort of 4 major software revisions. Version 1, unconscious and mass selection, moving to version 2, where we get an idea of inheritance, controlled genetics, to version 3 with marker-based methods and later on genomic selection. And now today, being in version 4 with tools such as genetic transformation and genome editing. And I want to emphasize here that all of these add on to each other. Genome editing is certainly useful for classes of traits and problems, but it doesn't replace other methods everywhere. But marker-based methods are known to be poorly suited for actually finding these causal loci, and for a number of different reasons. One, there's an ascertainment bias in the marker sets that are typically used to fit these models. They're a subset of the total pool of genomic variation that exists in populations. And further, you're only sampling the natural variation, and if your trait requires some novel, non-natural variation, then that's not an option or doesn't exist in these marker sets. And second, even if you were to sample all of the variation within a population, you still quickly run into this NP problem where you have a lot more variance to estimate effects for than you have observations in your breeding program. So the question becomes, what's the right causal locus funnel? How do we go from, for example, on the order of 83 million SNPs in elite maize germplasm down to an order of about 10,000 SNPs so we can start to either edit? My second question, going back, is how do we transfer knowledge across species? And so why might we want to do this? Well, in the future, we're going to potentially need to breed thousands of species. Historically, breeding has focused their efforts on the food yield traits in about 10 or so species. But with increasing mean average temperatures, some species, up to thousands, might need help being migrated to cooler climates. And we're going to need to scale efforts in those without investing the same amount that we have in these food yield crops. And so I'd like to know, how can we take all the effort and data sets that we've created in model plant species and transfer that knowledge into specialty and orphan crops? But it's known that these marker-based methods struggle to predict across distant populations, so we need new methods in order to actually do this transfer of knowledge across species. And so how do we do that? We can do that by looking at conserved mechanisms and using those to transfer knowledge. So the central dogma obeyed by all across the tree of life, things like chromatin that all eukaryotic genomes organize themselves into. And why should we use other species to train models? So first, more species is simply more training data. And the deep learning models that I'm going to present to you guys today require relatively large training data sets. And this starts to alleviate the n less than p problem. And if you can build models that can predict in held out species, this is a pretty strong validation that your model's learning some biology, because we know that functional sequences are shared across long evolutionary distances. And it also helps you detect when you're overfitting when you're in poor NP regimes. And finally, it helps prevent the memorization of retrotransposons. And because they evolve faster than their host genome, they're less likely to be shared across evolutionary distances. And this helps prevent the common problem with machine learning methods that sort of memorize that retro elements are common in regulatory regions and don't learn the real biology about those regulatory regions and just become transposon finders. All right, so just a quick overview of what I'm going to talk to you guys about today, or my PhD in a slide. So we're going to start with a DNA to chromatin model, move into an expression-based model that takes in the promoter and the UTR sequence, go into some bioinformatics work I've been doing to find the functional transcription factor binding sites from DNA, multiple sequence alignments, and finish off with a really wonderful collaboration with the folks at University of Washington, where we built a core promoter to expression model. All right, so DNA to chromatin. So I'm going to go through what chromatin is and how we commonly measure it. Why is it relevant for breeding? Why do I think that we can even predict chromatin state from DNA? And as a hint, we need both a conserved vocabulary and grammar, which I'll get into. Why are CNNs, the model I'm using here, well suited for this? How did I train them? And how well do they work? So we know that eukaryotic DNA is organized into chromatin. And so we can see here some cartoon representations of histone proteins that DNA wraps itself around. And when they're particularly compact, even though these DNA sequences have transcription factor binding sites, they can't be bound by transcription factors unless they're more accessible, and they can't then therefore regulate gene expression. So we measure chromatin accessibility many different ways, one of which is ATAC-seq. And basically, ATAC-seq measures where a massive transposase comes in and can cut, and it cuts preferentially in accessible regions. These cut sites are put on the ends of reads, and you can align them to the genome and call peaks from them. So these blue squares, for example, are binarized 1 or 0, accessible or non-accessible. Another interesting result recently seen in the grasses is that many of these accessible regions across tissues, almost all of them across tissues, are also DNA unmethylated. And this is really interesting because accessible chromatin is highly dynamic over developmental time. And so a lot of the peaks aren't shared across tissues. And if we want to build models of them, they have to be tissue somewhat specific. But if we can build a single model of DNA methylation, then we can take it in one tissue because DNA methylation is relatively stable over developmental time and only use one model to predict these regions. And I'm going to be referring to these peaks of lack of DNA methylation as UMRs, and they're peaks where there's a lack of DNA methylation in all three contexts. And just as a sort of example, you can see how consistent they are in this region across leaf and roots. So why should breeders care about accessible chromatin? Well, we know that they actually contain functional variants. So some prior work in the lab looked at the heritability proportion assignable to SNPs in coding regions versus accessible chromatin regions across about 40 or so agronomic traits. And what they saw is that SNPs within coding regions explain almost as much heritability as SNPs within coding regions, which we already know are important. So this has relevance. So can we predict chromatin state directly from DNA sequence? And why might we want to? Well, only about 5% of the maize genome is unmethylated. And so this would result in about a 20x reduction in the target space that we can mutate within, or should mutate within. And quickly, why DNA-based models specifically? So first, they are alignment free. And this is a common roadblock when you're doing cross-species analyses. How do you get good cross-species alignments? It can be very tricky. Second, some samples we can only assay with DNA sequence. For example, if you're collecting from an herbarium or other dead tissue samples, it's really impossible to do something like chromatin assaying on those. And nowadays, DNA sequencing is relatively cheap compared to things like ATAC-seq or ChIP-seq. Also, we have a pretty good idea on how DNA behaves over generations, so we can model reconstructed ancestors or predicted plant breeding program progeny and use those models to predict functional aspects of those non-existent organisms. And so it's this grammar between the transcription factor binding sites that should be a strong regulatory signal from the genomic background. And it could be helpful to think of these regulatory sequences of DNA in general as sort of a sentence. So the vocabulary are these transcription factor binding sites. So these colored rectangles I'm showing here. And the grammar is the spatial relationship or the co-binding between these motifs. And because the vocab is short and very degenerate, they occur randomly by chance throughout the genome. And just one TFBS is not really enough to say that that's regulatory. But if you suddenly see a lot of them together in a certain orientation, that's a better signal. But are these things conserved across species? We want to build across species models. We need to leverage that. So there's a reason to think from theory that the vocabulary is conserved at least across plants, because we know that the origin of a lot of these transcription factor binding domains predate the plant expansion. And so therefore the motifs they bind to should be highly conserved and common in plant genomes. And as a more concrete example, more recent work out of the lab trained Kamer binding models for about 100 or so maize transcription factors. And so they related whether or not a Kamer was more present in a region bound by a certain TF. And if you take all of those TFs and you build a tree based on the relationship between those Kamer weights, you can get this sort of tree that was built in maize. And what's exciting to see is if you start to color those tips by the orthologous transcription factor in Arabidopsis, the colors also group by the tree, suggesting that there is some conservation of that binding vocabulary across at least the angiosperms. So going back, we know that the vocab is likely to be conserved, or at least there's some evidence, but we really don't know if the grammar is highly conserved. And so that's my main hypothesis going into this work, is that the grammar is conserved and we can use that to predict chromatin state from sequence. So just an overview of the sort of classifier we're going to build here. Going from a window of DNA sequence, can we put it into a model and get some 0 to 1 prediction of whether it's inaccessible, accessible, methylated, or unmethylated? We already know that CNNs, the model I'm using here, are pretty well suited for this kind of task. What I'm showing here is results from 2015, one of the first CNNs, deep sea, to be applied to this in humans. And so we know that this is a good classifier because all of these black lines, which represent accessibility in different tissues, are far away from this diagonal, which would be what the random classifier would be at. And a year later, this other model, DAN-Q, showed that it can do slightly better across all chromatin tasks that I just showed previously. So these CNN models seem to be working. And also much more recent work in plants in 2011 or 2021 showed that they also do work well with high area under the curves close to 1. So just a brief overview of what CNNs are and how they work. So you can one-hot encode DNA as a matrix, and the convolutional layer in these CNNs, one of the first layers, is going to scan over in windows, and it's going to match these convolutional kernels to each window. And over time, it's going to learn what motifs are important to predict whatever your target is. And those, when it finds a match, is going to result in a high value that goes into later layers. And so this is well suited for this sort of like detection of motifs problem. And I use the DAN-Q model here specifically that I mentioned just previously because it also has this LSTM layer, which is a recurrent layer that allows you to learn not just whether a motif is present in a sequence, but also the co-presence of motifs and what their spatial relationships are. And this is exactly what I think should be a strong discriminator from genomic background to regulatory regions. But like I said earlier, these models are pretty large, and DAN-Q has about 1.6 million parameters, and it's a fairly small model for these days. So the data that I'm going to use to train it is chromatin data across the angiosperms from both monocots and dicots, so things like grape, common bean, soybean, and then your grasses like rice, maize, and sorghum. And I have ATAC-seq in about 10 or 12 of those species and methylation data in about 10 of them. In total, that results in about 312,000 ATAC-seq peaks across these species and about 360,000 UMRs. So not quite above P yet, but in a much better space than if we were to just train in one of these species. So how are we going to do this? I'm going to train the model in 2 different ways. One within species to compare to previous literature, and in another across species. So first, for within species, we'll take the peaks under 1 chromosome and assign them to our test set. We'll assign the rest to our training set. We're going to downsample so that there's a balance between our black and blue or accessible, inaccessible regions. And just so you can see now 3 to 1 to 1 to 1. And then we can get some performance metric for that model within the species on the test set. And we can do that for both accessibility and methylation. For the across species model, we'll assign the same peaks to the test set so that we can have an even comparison between the two. And then we'll assign training peaks in the other species, downsample again to balance. And now we have 2 bars that we can compare on the same Arabidopsis peaks for both accessibility and methylation. And so long story short, it turns out, yes, we can build models that predict chromatin state across angiosperms. So for example, I'm showing the methylation first. We can see that generally the orange and the blue bars are pretty close to each other, sometimes reversed. But the across-species models seem to be very competitive to models trained solely within species, even though they have to go across a longer evolutionary distance. And the same case is true for the accessibility, if not more so. What's interesting is if we have heavy sampling within specific clades, they seem to do a lot better than the across-species models, suggesting if we can fill out the tree a little bit more, we can do even better with across-species models across the angiosperms. And I also just want to highlight, we don't expect these cis-based models to be perfect for predicting chromatin state. We know that there are trans effects that come in from other areas of the genome that can silence these regions even without any change in the sequence underlying them. And I'll talk about those in a second. So what I asked next was, what region does the model do best or worst in? So I divided the genome up into either genic regions that overlap gene annotations, proximal regions that are within 2 kb of either the transcription start site or the termination site, and everything else is distal. And what I saw is that generally the model does best on accessibility in these proximal regions and worst on these distal regions. And it's a very similar case for the methylation models. So diving a little bit deeper, I noticed that a lot of the accessibility errors the model's making are false positives. So I'm summing to 100% within each of these columns. And you'll notice that, excuse me, about 70% of the predictions of the model that are open are actually labeled as closed in the data. So there's a high false positive rate. But I do want to emphasize here that this is not necessarily wrong, because the attack-seq peaks that I'm using were called with relatively conservative thresholds. And so if you have some certain peaks that are open in maybe half of the cell types or less in this data set, those might not have met the strength needed to be called in this data set, and they might be open, but the model doesn't know that. Or it suspects that, but it's labeled as closed and true. Okay, so some of the trans effects I alluded to earlier, one example of those would be, let's say you have a cis regulatory module sitting in a transposon, which is not an uncommon occurrence. And it looks like it should be a regulatory module by the sequence, but in reality, some host-directed, maybe RNA-directed DNA methylation might silence that transposon because of its context. And our cis-based model might not necessarily pick up on that. So how do we help it out? What if we started masking transposons? So we take in this case, let's say 600 base pairs of sequence, feed it into our model, and then it'll make a prediction. And then if it's in a transposon, we'll set that prediction to 0. And if it's not, we'll keep the original one and see how we do. And if we do mask transposons, we get significant improvements in both accessibility and methylation, suggesting once we start incorporating those trans effects into our cis-based models, we can actually get to more towards perfection. Next, I wanted to look at how well these models do across a number of different cell types. So I didn't mention previously, but this is leaf accessibility data. So the model's only been trained in one tissue type across species. And so I'll have one prediction for each of these peaks. And I wanted to see how well it does across a single cell attack-seq that has multiple cell types in it. So going from left to right on the x-axis here, we're moving from tissue-specific peaks to more generally accessible peaks. And we can see model performance follows basically how tissue-specific you are inversely. And I want to point out here that SC-ATAC is known to be relatively noisy. And so sometimes when you don't have many observations of a peak or you only have it in very few nuclei, it's hard to tell whether that peak is noise or a truly cell type specific peak. So this might actually not be predictable for the model. I also looked at genome-wide, how does performance differ across cell types, and it doesn't differ that much. Promisingly, the best performing tissue is the guard cell, which is a leaf cell type. That's great. And one of the worst performing is trichoblast, or a root hair precursor. So promising, but there's still not much of a difference. And the reason for this is either many peaks are open in very many tissues or very few tissues. And we know the model does really poorly on distal regions, which tend to be more cell type specific peaks. So across tissues, we're not going to do very well in those regions anyways, but it does very well in regions that are open in many tissues, and that doesn't change across tissues a lot. So that kind of explains why the model is sort of consistent across tissues. One of the common criticisms of deep learning models is understanding just what they're learning. They can make good predictions, but what biology are they learning, if any, to make those predictions? So how do we open up the black box, so to speak? One of the ways I did this was using a method from Peter Kruse' group called global importance analysis. and its sister method, positional global importance analysis. So the way that works is we're going to sample some background sequences just to control for potential co-binding motifs across the background. And we're going to embed some motif that we're interested in. So we can start testing specific motif hypotheses. And in my case, I took a database of known transcription factor binding motifs and just started embedding them in these random regions. And then you measure the difference between those set of embedded regions and the random background and just see how your model predictions differ. And it's the average difference between these that is known as the global importance. So how important is that motif being in the sequence to your model for making its prediction? You can embed this motif not just in the middle, but at any position and get a very position-specific effect of the global importance for embedding that motif. And so then you can start to rank these motifs by their effect size to the model. So in this case, I'm ranking them by their maximum positional global importance. So what motifs, no matter where you put them in the sequence, are very important for changing the model's prediction. And we can see that for the accessibility model, which had Arabidopsis held out predicting into a Arabidopsis sequence, it's paying a lot of attention to TCP family motifs. And this is cool because we know that some TCPs can be chromatin remodelers, so that's promising. For maize, there are some TCPs, but we also see these DOF type family transcription factor motifs ranked highly. We know that they're involved in seed and seedling development, no chromatin remodeling evidence that I'm aware of, but perhaps part of development is chromatin remodeling in this case. And across both Arabidopsis and maize held out methylation models, it's a much more consistent being these BBR-BPC motifs or AP2-ERF. We know there is some evidence for BBR-BPC to do chromatin remodeling, cool. And there is evidence that AP2-ERF, or they're known to be involved in development, but the case is less clear. So in summary, I've shown that these recurrent convolutional neural networks can seem to model chromatin accessibility and DNA methylation directly from sequence and across plant species, and that these across-species models are competitive with within-species models. The accessibility A to Z models seem to perform best on gene-proximal and commonly accessible regions. And incorporating trans effects starts to reduce our models' cis-based models' false positive rates. And finally, these TCP family motifs seem to rank highly in global importance to the across-species accessibility models, and it's a much more consistent case being AP2-ERF or BBR-BPC for methylation. All right, so moving right along to our expression model, or my expression model, I guess. So one of the motivations for making this model was to try to move towards some more efficient promoter bashing. And so promoter bashing is where you'll take random guide targets upstream of a gene and just start trying to make mutants until you see the effect that you want. And they're untargeted, but what if we can build a promoter model that can instead reduce the space that we would need to randomly bash from on the order of 2,000 base pairs to more like 100 or so. So plenty of our human expression models already exist in the literature, and I'm going to test 4 of them here. The first is FNet compression, which is a relatively tiny model at 60,000 parameters. Hyena DNA, a more recent one at 450,000. DanQ, we're familiar with at 1.6 million. And Informer is currently state-of-the-art at about 250 million parameters. I couldn't quite get Informer to run on the hardware I had available, so I downsampled it to about 50 million and called it Miniformer. So previous work in the lab also showed if you have coding sequence going into your model, then there's a potential for that model to start to memorize gene family. And so in this case, if you have things that are looking like transcription factors, for example, in their coding regions, then the model will just tend to think that, oh, if you're a transcription factor, you tend to be lowly expressed versus something that is more highly expressed, and not learn anything about the cis regulation differences between the two that actually explain expression. So there's a couple of different ways to improve existing expression models. The first, as I mentioned previously, within species models have limited independent training observations. Second, if you don't mask your coding regions, then that can enable overfitting on gene family. It's also been seen that there's a poor recall of these models on rare EQTL alleles. For example, a lot of these expression models just fail to detect even high effect EQTL if they're not present at high frequency. And there, as of today, well, before this work, there was no level comparison between all of these architectures. So it's hard to tell whether this model is better because it had a different data set or the problem was easier. And so I wanted to make sure that we can test everything on a level playing field. And so the data I'm using to train these models are 32 new long-read genomes and RNA-seq data from wild relatives of maize. And these genomes are really the result of collaborative effort between Arun Seethram and Michelle Stitzer, along with collaborators at the Danforth Center, Taylor and Toby, who actually did the sampling of these species. And it spans the Andropogonea tribe, of which maize and sorghum are a part of. And so the schema for this model is I'm going to train on the relatives of maize and try to predict in maize. So very similar to the last model, but also following the advice from the previous lab work, I'm also going to do the orthogroup guided splitting. So in this case, I'm going to have a holdout set of genes that are in different orthogroups from my training set and also in different species. So this is a very difficult problem. In total, I'll have about 1 million training observations for these expression models. So in my model, I'm going to start at the translation start site and go 1 kilobase pair upstream of the translation start site and try to predict the expression levels either in specific tissues like leaf, stem, or flower, or the maximum expression across tissues, or the binarized form of that on-off expression in any tissue. And I want to highlight that the within-gene performance is not necessarily equal to a cross-gene performance. And a cross-gene is typically what's been shown in the literature. Excuse me. What I mean by a cross-gene is if you take a look across all the genes in the genome and all the species that you have in your training or test set, if you can correctly separate the highly expressed genes from the lowly expressed genes, it doesn't mean that you have the ability to distinguish between, for example, closely related orthologs. So it's classic Simpson's paradox situation where you looks good between groups, but then you start to go within groups and your correlation levels drop. So first, just to compare to existing literature, the across-gene regression results are about equal to a within human benchmark. What's cool to see is one of my best performing models on one of the best performing tasks, maximum expression, is about 0.52, and it's DAN-Q. And the within human data, Informer, gets to about 0.57. So we're almost there, not quite, but these models are predicting across species in this case. I also compared it to the previous work from the lab that did this within maize. So for the on-off expression model, one of the best models is FNet compression at an AU-ROC of 0.77. And this is exactly equal to the model that was trained within maize that took in only promoter regions. And I want you to notice there's also a slightly better model that took in both promoter and terminator, and we'll get into that in a second. But first, the within-gene performance dramatically drops once you start looking across orthologs within maize. So I was showing cross-gene correlations of on the order of 0.5, 0.6, but within genes across different orthologs, the average correlation drops to about 0.1. And there's not really much difference between even models of several orders of magnitude parameter difference. And it's only slightly better on average on the maximum expression task. And this agrees with the same benchmark that looked at this in human on Informer, the current state of the art, in pattern. So looking at one of my best models, DAN-Q, again, just to sort of emphasize how poorly it ranks these orthologs by abundance, I'm looking on the x-axis here, the predicted expression change by the model between any pair of orthologs on the log 10 scale. And then we're looking at the observed expression change. And generally, it's just not doing well for anything except things that are more towards the tails. So until you get to about 100 or 1,000 times X expression increase, it's not able to differentiate. Perhaps the model's overfitting quickly on these huge expression differences and never really gets a chance to finely distinguish expression. Another thing that might be going on is the previous work in the group showed that really it's the 3' UTR and the terminator that was needed to distinguish between at least maize orthologs. And so potentially what the model needs is to also include that sequence. But I also want to emphasize again that if if we do this, future work really needs to make sure to mask those coding regions so it doesn't memorize gene family. I also looked at where the Dan-Q model was paying attention for predicting expression levels in the max expression task. And what's cool to see is it actually highlights the core promoter. So note, I didn't give the model the transcription start site. I started at the translation start site, and I've sort of manually aligned these important scores for you to the transcription start site, and it highlights across all the maize genes the core promoter, which is, we know, is important for expression. Yeah. Okay. The other thing I looked at is it's a common refrain in deep learning that we need more training data, more data, better models. And I wanted to know if that was actually the case, because that data doesn't come for free. So I took various subsets of my training data, starting at very small fractions of it and gradually increasing it to the 1 million number of observations. And I do see the performance between the 500 and 1 million is still increasing, suggesting there is a need for more training data depending on the price point at which it costs to acquire it. So in summary, it seems the current genomic deep learning architectures perform well across distantly related species, and this agrees with within human benchmarks in both pattern and magnitude, and it has nearly the same performance as maize models without seeing any maize sequence. They don't seem to be sensitive enough yet to rank closely related sequence, which agrees with the human benchmark as well. Perhaps we need 3' UTR, but I also think it's interesting to think about if the 3' UTR is a region that evolution has happened to favor to fine-tune expression, or if it is actually needed to fine-tune expression. So the third point is that the models do seem to recognize the core promoter as important across all genes. That's a good validation. And it seems we could use some more training data to saturate the performance of these models. All right, so moving on to a more bioinformatics-focused approach, looking at multiple sequence alignments to predict functional transcription factor binding sites. So why functional transcription factor binding sites? Well, compared to an accessible chromatin model, if we can actually find the real binding sites, that would be even a more efficient editing target, because not every base pair in an accessible region is necessarily a binding site. And I kind of alluded to this before, but motif scanning, if you just take known motifs and scan them across the genome, there's a high false positive rate. So if you look at any random promoter upstream, you're just going to see a lot of hits and they overlap, and there's a lot of them, but how many of them are actually functional? Which ones are functional? And this is so bad that these authors kind of exasperatedly conjectured in 2004 that this futility theorem means that about 100% of your predicted binding sites are non-functional. 3 orders of magnitude difference. But we can use other information to filter down these methods, such as evolutionary conservation. So if you do your motif scanning as usual and you detect a region of the genome that's more conserved over time, that's more evidence that these are actually doing something. And this has been known for a while as phylogenetic footprinting. But why use phylogenetic footprinting now? What's changed? So first, there's new aligners on the scene like AnchorWave that can handle these plant intergenic regions much better than previously. And this is important where a lot of these non-coding, in these non-coding regions, there's a lot of regulatory regions that we want to study their conservation. Also, I mentioned we have 32 new high-quality long-read Angiopregoniae assemblies. And we also have 400 new short-read Angiopregoniae assemblies to even increase the depth of these MSAs further. And so all of these assemblies came from the NSF Pan-Am grant, which collected about 800 Angiopregoniae samples worldwide. As I mentioned previously, colleagues at the Danforth Center, Taylor Abishan Elder and Toby Kellogg, run around the world and collected Angiopregoniae from fresh tissue samples, or either went into herbariums and silica collections to get these samples. And so if you build a tree out of all these samples and you sum along all of the branches in that tree, they sample approximately on the order of about 1 billion years of evolution, which comes out to about 30 to 50 mutations per base pair. So a very powerful data set to measure constraint. And so if you look at these short-read assemblies, which are thanks to the efforts of Twee and Cinta for the sequencing and Amy and Charlie for the assembly, if we look at the BUSCO completeness of these assemblies, generally they seem to be fairly complete. And if the genes aren't complete, they're at least present in a majority fragment. So fairly contiguous. And so as part of the long-read assembly work, a collaborator, Armin Shivan, took the long-read assemblies and used the cactus genome aligner to build a multiple sequence alignment between them across all 32. And then he used FastCons to do that phylogenetic footprinting I talked about earlier and find the conserved non-coding sequences, or CNS. And in maize, there's about 95,000 of them that he detected. So now my work comes into adding in those short read assemblies to see if we can get further depth in these alignments. So starting with a species tree that contains the non-ref, which I'm referring to the short read assemblies as, and the ref, the long read, we can align the non-reference contigs to the nearest reference assembly so we can avoid some of the reference bias, and then use that cactus alignment to lift over all of these alignments into the same MSA space and get MSAs of these conserved non-coding regions that have hundreds of Angiopogon A assemblies in them. What I noticed looking at some of these CNS regions is that only about 60,000 of those are widely covered across the Angiopogon. There are a subset of them that are only present across a number of the long read assemblies, and we sampled heavily in the zeoclade for the long read assemblies. And I wanted to focus this analysis on things that were present across everything, so I decided to filter out to this upper right quadrant here. So that brings me down to about 60,000. The next thing I did was use the Jasper transcription factor database to scan for those motifs with the FEMO tool. And so looking at a real example of an alignment within one of these CNS regions, we can imagine that there are some motif hits within it right here. And I'm going to use FAST, so the same set that contains FastCons, to test whether these motifs as a region are more constrained than the bases around them. And I'm going to do some very simple filtering to get rid of these very gappy columns. And so when I do that and run the test, I get about 56,000 transcription factor binding sites in maize that are constrained to the Bonferroni corrected p-value across pan-end. Okay, so the next thing I wanted to do is look for binding preferences in these across these motifs. And so to do that, I did an all-by-all alignment of the constrained motifs that I detected, also along with all the Jasper motifs from the database. And so since some of these motifs are shorter or longer than others, I took the longest motif and I slid the shorter one across in all possible positions and took the minimum of the Euclidean distance between the two and used that as the alignment score. And it has to be a Euclidean distance. This is a common motif comparison metric, but also remember these motifs are actually matrices and they represent frequencies of bases at each position and not a single base. So at the end of this, I have about 58,000 by 58,000 distance matrix with all the constrained motifs in Jasper. And that can be a little hard to visualize. So I used UMAP to reduce the dimensionality and was actually very pleased to see that they cluster by class and family according to the Jasper labels. So that's a good validation that the alignments actually worked and are doing something. And so let's see if they have some specific binding preferences within them. So I looked in one class and family, in this case, the BZIP group Bs, and I clustered them according to their motifs. And I see that there is this big cluster here that has a Jasper motif within it. But there's also two clusters down here, the pink and the red, that don't have any annotated Jasper motif, which is cool. It suggests some novel binding pattern that we haven't seen. And when we look at the motif preferences in each of these clusters, there are actual differences. For example, between the pink and the red, there's a strong CA preference versus a strong CG preference, or a strong G preference in the red versus a very weak T or A preference up here. So certainly interesting to see, but I'd like to get more validation. And so I looked at how the subcluster presence or absence correlates to expression or explains expression variation. So I'm doing a very simple linear model for this expression testing. So the Y values are going to be TPM, or transcripts per million, a measure of RNA expression, log 10 transformed. And the X matrix is either the presence or absence of a motif from that subcluster and is it within 100 kb upstream of a given gene, which is well within what we know cis-regulatory regions are capable of interacting with the core promoter. And so I have about 5,000 genes that have any one of these motifs upstream, and there's about 1,600 motif subclusters. And what I'm going to do is for each of these columns, for each subcluster column, I'm going to randomly permute and measure the change in R squared of that model. And I'm going to do a couple replicates of permutation and measure the average change in the model's R squared by permuting that column. And so focusing on the right side here, every row is a replicate RNA-seq experiment, and we have multiple replicates per tissue. And generally, the R squared values don't differ very much, ranging from 0.2 to 0.3. And along the x-axis here, we have those different 1,600 motif subclusters, and I've normalized by the R squared of each model, the delta, so that we can compare across rows. Now, this can be kind of hard to visualize, so let's zoom in just a little bit to again, the B-zip factors and the C2H2 zinc finger factors. And we can see some cool initial patterns. So for example, we have this subcluster that seems to explain a lot of the variation in endosperm tissue expression, and it shows up across both tissue replicates. So that's a good sign. We have some subclusters that seem to show up in anther tissue as well. And then some subclusters that don't seem to be specific to any tissue in particular. So for this section, to summarize, I've seen or identified these constrained maize transcription factor binding sites, and they seem to cluster into subfamilies that have very specific binding preferences. Some of them are not annotated in known TF databases, so novel motifs. And the upstream presence or absence of these constrained subfamilies seems to be important to tissue-specific expression patterns. So to close us off, I'm going to talk about just a wonderful collaboration with some fine folks at the University of Washington, where we built a core promoter to expression model. I've talked a big game about these models, saying that we could use them for genome editing, but let's prove if they can actually work or not. So what Toby and Jackson did in Christine Queisch's group is a star-seq assay. And glazing over a lot of the molecular biology, just you know that you can synthesize any 170 base pairs you want, put it upstream of a reporter gene, put it into either tobacco leaf or maize protoplast, and measure the relative expression to some baseline. And what we did in this work was take 170 base pairs upstream of the transcription start site in Arabidopsis, maize, and sorghum. And I'm calling that the core promoter. The good news is star-seq is replicable within the expression system. So looking across replicates 1 and 2 in tobacco leaf, the same promoter agrees with an R squared of about 0.91. When we look in the maize protoplast, it's a little bit less at R squared of 0.79, but still pretty agreeable across replicates. And what I want to emphasize here is if we start to compare the same 170 base pair promoter across maize and tobacco systems, they only agree with an R squared of 0.31, really emphasizing how much of the variation expression depends on the cellular environment versus the cis promoter sequence. So my contribution to this work was building this bidirectional CNN model to predict core promoter strength. So what's bidirectional about it is typically these convolutional neural networks scan along your input sequence only in one direction. They don't understand DNA strandedness. So a modification I made was to have the model automatically flip the motif into its reverse complement and scan it along the other strand all at once, so that it can use the same kernel to learn both the forward and the reverse strand instead of wasting two. And that's great because it results in a more efficient use of parameters, which is important if we're limited in data quantity. And in one of the most exciting moments of my PhD, we found that these models actually work. So we took both 3 SNPs suggested by the model or 10 SNPs suggested by the model and actually put them into tobacco or maize and show that they increased expression levels significantly. And this doesn't matter whether you're in tobacco or maize, or if you have a 35S enhancer upstream or not. It increases expression in all cases. And not only that, the model is able to predict the degree at which expression increased. So they do seem to have the ability, if you can control the trans environment, to predict across different alleles very closely related alleles. So in summary, these CNN models seem to be highly effective learners of cis promoter variation if you can control that trans environment. And the suggested edits from these models actually work in Planta. And it seems that these could be the way towards more efficient promoter bashing. So bring us all together again. The DNA to chromatin model we saw does work across species and also is comparable to within species trained models. So great. For the promoter and UTR expression model, it does work well across species, but it still struggles to predict across different orthologs. So I'll give it a yes and a no sort of success there. There's still some more work to be done to validate these subclusters in the transcription factor binding site work, but it's showing some interesting patterns between subcluster presence absence and expression. And finally, the core promoter expression model not only seemed to learn the relationship, but also then predict edits that could make changes, and actually those changes reflect what the model predicted. So I'll give that 2 checks for super success. And with that, I would just like to acknowledge my funding. So from the NSF and the GRFP specifically for my first 3 years of graduate school, and USDA ARS for the rest. I've, as you can imagine, had to leverage a lot of different compute across the years, from Exceed and the TAC cluster to the Frontera system, and more recently on the USDA SciNet cluster and BioHBC. Thank you very much. Of course, my committee, Ed, Amy, and Eric, for your advice, and Cornell for having me. I'd also like to thank X for just not only making my internship a very valuable experience, but just plain fun. My co-authors, without whom, of course, I could have just not done this work. The Buckler Lab and Synapsis for both the support and all the good times over the years. And finally, thank you to all the open source software developers and packagers who have probably spent countless hours making the tools that we rely on, and also the packagers for making them just easily installable for us. So with that, I'm happy to take your questions. Yeah.
You can go back to your thesis on a slide there.
Yeah.
So the upper right-hand corner is, I guess when you're trying to predict within orthologs, you can predict the amount of expression across orthologs pretty well, but not between genes within orthologs, right?
Yeah, so I'll be a little bit more precise with my terminology there, sorry. So when I say ortholog in this case, what I mean is across one gene in maize across different alleles in the NAM population. So not necessarily 2 paralogs within the maize genome. So sorry if I wasn't clear about that.
And the training that you have is the total expression of that gene across many orthologs, right?
It is either the max expression of that ortholog across all tissues that I have data for, or just one single tissue expression value.
Could you try to train on the deviation within a allele? So for an allele, it's deviation from the mean for that gene. Does that make sense?
Yeah, you could absolutely do that. And the previous work, I didn't highlight what they did there, but they did exactly that. They put both ortholog sequences in the model and tried to predict the expression, and it does really, like, the expression change, and it does well. But it doesn't, like, for example, take a difference in DNA sequence. It takes both in the input. Yeah.
Yeah. Do you know roughly how differentiated those 2 orthologs are from one another? Why the Washburn approach is easier than this one?
No, I don't know the sequence number off the top of my head.
Do you know? So I mean, they're about 8 million years diverged.
Oh, I see.
Okay, so that's an easier problem than this.
Right.
Differentiating one SNP causes an expression difference.
So in their case, orthologs were across maize and sorghum. In my case, they're across like B73 and Mo17. Are there any questions online? I can't really see the chat. No? Cool. Yeah.
Did you ever look at transferability across tissues for the DNA to chromatin model?
And you do a model?
Transferability across tissue. So I did show like sort of you mean performance across different tissues, like the single cell work?
Well, like if you train in leaf, how well does it do in root?
Yeah, that's a great, and if I had the data set to do that, I would have, but at the time I did the model, I only had the leaf accessibility across that vast number of species. So no, I think it's an interesting question, but I didn't have the data at the time to test that. Yeah. Call. Ooh, should come to my defense now. So yeah, I've just been sort of roughly saying core promoter, like anywhere from 50 base pairs to 150 base pairs upstream. Promoter is such a wide number. I've seen up to 2,000 base pairs from a gene be called the promoter. So yeah, it's a very loose term and I'm abusing it, sorry. Saren, I think, yeah. Yeah. Yeah, I mean, it highly varies based on really genome size. So like in Arabidopsis, things on average are going to be a lot closer together than in bigger genomes like maize, where things are pushed out by a lot of this transposon activity over time. And so within the grasses that can tend to have larger genomes, that range can get up to 100, 250 kb on average. So it really seems to depend on genome size. Yeah. Oh, John Luke.
You train these models on data.
Yeah.
And sometimes it's accessible or not, methylated or not, conserved or not. But it's like you had a comment at one point where it seemed like accessible or not, it's actually not binary in biology, right?
Yep.
There's some continuous nature of accessibility.
Yeah.
So it's a general question about statistics maybe of like what do you gain and what do you lose when you convert something that's in some ways continuous to something that's binary for training a statistical model?
Yeah, I mean, that's a great question. And part of what you gain is a high false positive rate. I mean, it's something that we.
So that's a loss, right?
Do you like my phrasing? Yeah. And yeah, so there's more, as we get more and more single cell data, we can ask that question, like train a model on the proportion of a given cell type that's actually this peak is accessible and you know, we had the data we had at the time, and I think nowadays we can certainly make a more quantitative model for accessibility. Yeah, because I totally agree. Biology is not in this case.
Your answer suggests that usually if you convert to binary, you lose something. But you had a sort of scatter plot where I think it was also within the promoter to expression where you said, oh, maybe it's overfitting on these things that are massively expressed, that have massive fold changes.
Yeah.
And that made me think, oh, well, why don't you just make that one binary and then it won't know which ones are have massive fold changes.
Yeah.
Which that might be a case where you gain something, but maybe I'm wrong. I mean, comments?
No, I think that's interesting, and some potential ways to think about that is first training a model on pseudogene or not pseudo. When I say pseudogene, things that are expressed or not expressed in any tissue, and then have a model that fine tunes on sort of expression level, so you don't have to learn both any expression all at the same time as you're trying to learn expression fine tune. And I think another problem. Why I emphasize the tails as sort of overfitting is because I used the mean squared error as my loss function, and maybe that's weighted much higher in my model and just learns that. So,
Thank you.
Yeah, sure. Sam.
Do you think these models can be improved by taking advantage of different architectures, or do you think it's more of a problem with lack of data?
I think it's a lack of not only data, but sort of well replicated and controlled data sets. I mean, I tested model architectures that had vastly different orders of magnitude differences, and they just really didn't show much performance differences. So yeah, from what the evidence I've seen, I think we just need better data, more controlled data sets. And if someone has a really cool architecture they propose is the best, and test it against that data set, just like we have these CASP competitions for protein folding, and that's how we knew AlphaFold was one of the best performers, because it didn't see any of the data beforehand. So I think having these sort of holdout, well-curated data sets is really essential moving forward. So
Travis, I was wondering, when you're fitting your transcription factor binding site models, you're only using at the end 5,000 genes. Why, why is that, rather than like the 20,000 or 25,000 genes?
yeah, so good question. In this case, I only chose genes that had at least one motif of the subclusters that I identified upstream. I could certainly add in the other 20,000 that don't have any, but there would be a lot of zeros.
So there were that many genes without a motif?
Without a constrained motif that I could detect with my MSAs, yeah. Any last questions?
Okay, Travis, we ask
could
you more questions.
Perfect.
Well, thank you very much.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.